I'm very happy that I can introduce shortly somebody I don't have to introduce a lot. It's Dr. David Livermore. I think you all know David. What should I say? He's an expert, to be humble, in beta-lactamases and gram-negative resistance et al. And David agreed to give the introductory talk at the symposium, and we'll talk about extending the scope of resistance mechanisms by MALDI TOF. So David, we are all quite curious about what you can tell us.
Well, Mr. Chairman, ladies and gentlemen, many thanks for the introduction. Many thanks to Bruker for the invitation. It's embarrassing to be a microbiologist, isn't it? You look at virology, clinical chemistry, they've undergone paradigm changes over recent years. The speed is incredibly more than it was half a century ago. But the general speed of medical bacteriology proceeds more or less the same as it did in Alexander Fleming's time. Here's our unfortunate patient, 24 hours to grow a culture from the urine or the blood or whatever we've taken, and another 24 hours to get the susceptibility of whatever's been grown. The only real acceleration has been in identification, but the identification still has to await the susceptibility test result. And meantime, our unfortunate patient has to be treated empirically, based on guidelines, based on probabilities, based upon statistics. And getting it right is critical, at least in the severely ill patient, because there are multiple studies that show that if the empirical treatment given to the patient transpires to be inappropriate, transpires to be inactive against the pathogen, then in severe infection, the mortality risk is roughly doubled. And there's a whole array of studies here: appropriate therapy, the antibiotic given empirically turned out to be active against the pathogen isolated, and contrary case, the pink, where the empirical antibiotic proved not to be active, illustrating what I've just said, that mortality roughly doubles. Now, many of these deaths are because the antibiotic simply doesn't cover the whole group of pathogens. Patient was given, let us say, ceftriaxone and actually had an MRSA or a Pseudomonas, which ceftriaxone never would nor could cover. But others are because the bacteria had specific resistance. And there's an array of studies here and a meta-analysis by Schwaber and Camelli showing that if your bacteremia was due to an E. coli or a Klebsiella that had an extended spectrum beta-lactamase and therefore was cephalosporin and very probably cross-resistant to quinolone resistant, then your chance of death, again, roughly doubled. So if only we could identify pathogens and resistances quicker, we could tailor patients' treatment quicker as well, and that would aid the individual patient, more likely to get appropriate treatment, and it would be better for antibiotic stewardship too. What can we do to speed up identification of pathogens and resistances? Well, there's lots of interest at present in terms of PCR and even sequence-based detection of pathogens and their resistances directly in the clinical specimen. Some of you were perhaps in the previous session where we were talking exactly about that. But there's other methodologies that might be applied too. Some we've been using for years. Rapid colorimetric tests to detect beta-lactamases and immunological detection of PBP2A, PBP2' to take at least the primary culture and answer the question, is this MRSA? Isn't it MRSA? And we could add to the list there plating of original clinical specimen directly onto colorimetric media that detect presence or absence of resistance methods. But I mentioned already the increasing use of MALDI TOF to identify pathogens swiftly from the primary culture within minutes, rather than, as we previously had to, having to wait 24 hours for the API or whatever to give the identification. Can we adapt MALDI TOF to detect resistances? Now, I've agreed to give an introduction. I don't for a minute claim to be a personal expert on the assays that I'm going to outline. But what I am going to try to do is to review what's been published, the ideas that people have come up with. Many, I'm going to say, need more work, need refinement, but hopefully I can point you towards a whole series of studies which suggest that there is potential here. For anybody not familiar with what MALDI TOF is, here we've got the specimen in a matrix, we bombard it from a laser, ions are generated, the ions then migrate towards the detector, their rate of migration is determined by their size. Smaller ions go quickly, bigger ions go more slowly. So we sort by molecular mass. Think then of a beta-lactamase. A beta-lactamase hydrolyzes the beta-lactam molecule, and that hydrolysis introduces water. So the molecular mass goes up by 18. All did chemistry at school, yes? Oxygen 16, 2 hydrogens, 1 each, is 18. Molecular mass goes up by 18, and this hydrolysis product may then fragment or not. But in any case, molecular mass of a substrate changes. So, a lot of workers have done this type of assay. Taken primary culture plate, resuspend the bacteria, add an antibiotic solution, incubate for 2 to 4 hours, and then put the sample through the MALDI TOF and see whether or not the antibiotic has been changed by the bacteria. Some have used intact bacteria, some have used bacterial lysates. Here's one group's results, Rabat and his colleagues. No bacterial extract added. There's the meropenem peak, and I think it's 384, I can't read from this angle. And there, with the meropenem incubated with carbapenemase negative bacteria. So, same pattern. But add E. coli with an NDM carbapenemase or Acinetobacter with an NDM carbapenemase, you get new peaks appearing which correspond to fragmentation products of the initial hydrolysis product. You'd expect something 18 up from meropenem. You don't get that. You get the fragmentation products. Different group here, but the same sorts of results. Here is the matrix, no imipenem, now with the imipenem added, and then with an Acinetobacter baumannii, which was sensitive, I think, to carbapenems, didn't hydrolyze the imipenem at all, so still just the imipenem peak, but now with an imipenem-resistant Acinetobacter baumannii, the imipenem peak has disappeared, and then the fragmentation product peak has appeared. And the same second group of workers actually tested bacteria with a wide array of different carbapenemases, either incubating the bacteria with the antibiotic for 2 hours or 4 hours. And with the longer incubation, all the ones that were known to have carbopenomases gave a peak for the fragmentation product of the hydrolysis product, whereas the controls with just an ESBL or the carbapenem susceptible acinetobacters gave no hydrolysis product. So good evidence there that the MALDI TOF can be adapted to a beta-lactamase assay. The very recent work from my colleague and friend Justin O'Grady here, looking at 2 different groups of carbapenemase producers. All these that are labelled in white have a KPC carbapenemase, and each of them strong reaction after 2 hours, let alone 4 hours. This is with ertapenem now and lots of hydrolysis product. The ones that are flagged in blue have OXA48, a much weaker carbapenemase, and very little hydrolysis product detected at 2 hours, more at 4 hours. So perhaps by the strength of the signal, one can get some guide to the type of beta-lactamase, and although I haven't included the slide, you can strengthen that view based on adding inhibitors. EDTA will inhibit a metallocarbapenemase. A boronic acid will inhibit a KPC, and not much inhibits OXA48. The purple background there are the carbapenemase negative and carbapenemase positive controls. Here's one that's counterintuitive. Again, it's Justin's work. What we did was to take a range of KPC positive Klebsiella graded by ertapenem MICs. So the blue is the magnitude of the MIC. We thought that that probably reflected amount of KPC enzyme. So we thought we'd get more hydrolysis product with the one with the highest MIC. In fact, with that one exception there, we get the reverse pattern. The least hydrolysis product with the highest MIC. And what we now on with investigating, and this is work in progress, is whether what the real case is all these have similar amounts of KPC, but these are less permanent. But either way, relationships between type and amount of enzyme being teased out. Can MALDI TOF be applied to other drug-modifying enzymes? Well, so far as I can see, and I stand to be corrected, nobody has done an assay to look clinically for amino glycoside modifying enzymes. But this group, Green and his colleagues, did a study on the substrate specificity of aminoglycoside acetylating enzymes. Now, normally an acetyl transferase will put an acetyl onto a free amino of an aminoglycoside, and that increases the molecular mass. What these people were interested in is whether you could get these enzymes to use propionyl-CoA instead of acetyl-CoA. So a 3-carbon chain instead of a 2. And yes, you can see that the fingerprint, we get an extra peak here, is changed. The enzyme is capable of making propionyl neomycin when it's given propionyl-CoA. So it's possible then to change to utilise this type of methodology, surely, to do assays to see what aminoglycoside modifying enzyme an isolate might have. Note though, one problem is that they looked at 2 different acetylating or propionylating enzymes, modifying the neomycin at different positions, the 6' or the 3', and you get the same pattern from either because the molecular mass of the product is the same either way, though the specific position change is different. Another completely different sort of modifying enzyme are those that change the ribosome in such a way as to block drug binding. Now there's a range of these. This is CFR, which modifies 23S ribosomal RNA so as to block the binding of linezolid, chloramphenicol, clindamycin, but there's others, for example, which change the 23S ribosomal RNA to block binding of macrolides and clindamycin, the determinants, and there's 16S ribosomal RNA methylases, which change the ribosome to block the binding of a wide range of different aminoglycosides. The group here showed that with the CFR strain, they extracted the ribosomes and did MALDI TOF, they could get an additional peak and the disappearance of a normal peak. Now, this wasn't really convenient for a clinical lab type assay because there was a need to purify the ribosomes enzymes to digest products to give the fingerprint that I illustrate. But there is a potential perhaps to take such an assay, to convert it to kit and microfluidics form, and then to make it more convenient. And that would be useful because these ribosome modifying enzymes are a growing resistance problem. I move now to what I would say are a series of interesting observations, all of which need further work, but may give pointers, again, to how MALDI TOF technology might be used to further investigate resistance. This is work from Australia, where, curiously, they have a problem with Van B Enterococcus faecium, but no Van A Enterococcus faecium, and they plate specimens onto agar containing a low level of vancomycin and try on that medium to distinguish what's got Van B and what hasn't. Classically, that would require additional testing, but what was done was to take colonies growing on this agar, to put them through MALDI TOF mass spectroscopy and to look at the profile. And the green is a sort of average profile for those that had Van B, versus red, which is an average profile for other Enterococcus faecium that turned out not to have Van B by PCR, but somehow struggled through on the median. And you can see a distinguishing peak there. And here, again, they looked at some Van A controls sourced from abroad and a difference in pattern between the Van A and Van B. Very good sensitivity and specificity reported, and it's surely tantalising. Is this a convenient way of detecting and distinguishing Van A and Van B enterococci on a primary culture plate? Or is it that Australia has particular clones of enterococci and it's clone rather than resistance mechanism that's really being distinguished? Does it work for Enterococcus faecalis? What on earth were these faecium that didn't have Van A or Van B that managed to grow on the median? And what somebody urgently needs to do is to take an Enterococcus and put in Van A and Van B determinants so we have the same strain with and without the resistance mechanisms. And is the profile the same or is it different? Great experiment for someone to do. Lots of authors have looked at MALDI TOF as a means of distinguishing methicillin resistant and methicillin susceptible Staph aureus. And I summarise the results here. Some succeeded, some failed. Candidly, I would say it's complicated compared with looking for PBP2' by a latex agglutination. So for MRSA detection, there's probably easier methods. But look at this. This is work by Paul Myshik and his colleagues, starting with a tycoplanin sensitive Staph aureus, which in the patient yielded a tycoplanin resistant mutant. Put through the MALDI TOF, an additional peak appears. It then reverts and the peak disappears. The association is clear, but it's one strain. Again, more work is needed. A rapid method to identify tycoplanin non-susceptibility in staphylococci is potentially interesting. And Robak, who elegantly summarised a lot of the work that's been done on MALDI TOF for resistance detection in a review earlier this year, highlights a series of other cases that might be explored, particularly looking at cell wall components. Lipopolysaccharides. Lipopolysaccharide structure is a correlate of polymyxin susceptibility or resistance, looking for specific, very populous porins in the outer membrane. They determine permeability. And remember a little while ago, I was saying, well, a possible explanation about getting less hydrolysis product from ertapenem with the more ertapenem resistant strains tested as intact cells was that they were less permeable. That surely needs exploring. There's been some interest in so-called mini-sequencing of DNA, though it's likely that straight sequencing of DNA is becoming so much faster and more convenient that this perhaps won't be the way to go in this particular aspect. I wanted now in the last couple of minutes to talk about a more general issue. And this applies whether we're talking about detecting mechanisms by MALDI TOF, by PCR, or by next generation sequencing. They are all predicated on speed. Getting a result to indicate a resistance mechanism rapidly from primary culture, or in the case of DNA technology, perhaps directly from the clinical specimen with no culture at all. But they are going to find a mechanism or absence of a mechanism. They're not going to find, as we've talked of them so far, an MIC, whereas needless to say, both CLSI and UCAS are fixated upon MICs and the relationship of MICs and to pharmacokinetics as a predictor of clinical success or failure. But is there just possibly a way to balance that conflict? Because make no mistake, it is a coming conflict. Molecular susceptibility testing will tell you mechanism, but it won't tell you MIC. But this is now with fungi, with Candida albicans. And this group took a range of isolates of Candida albicans that varied greatly in fluconazole MIC. And they incubated heavy suspensions of these yeasts together with fluconazole. And then they shot the samples through the MALDI TOF. And what they found, and this is for an isolate with an MIC of 2 mg per litre, then the profile they got changed around MIC. And that held good for isolates with a wide range of different MICs. Which just begged the question, would it be possible to devise for bacteria as well some rapid susceptibility tests based on using heavy inocular and looking at changes in profile? Particularly, for example, when the antibiotic induces filamentation of the bacteria as a primary response. Well, ladies and gentlemen, what I hope I've managed to do is to stress the point that there is an urgent need for the acceleration of microbiology. In a world where we have to deal with more resistant bacteria, where we recognise that in the severely ill patient it's critical to get primary treatment that works, we really cannot go on as we have with a 48-hour lag before we know bacterial susceptibility. We've got to look to more rapid methods, be it colorimetric media, be it PCR, be it next generation sequencing, or be it inventive ways to use the MALDI TOF. And what I've tried to do in this opening presentation is to outline how the method certainly can be used to detect carbapenemase activity. There's lots of papers on that now. The methodology is clearly established. But it could potentially be used more widely. Aminoglycoside modifying enzymes, if the tests can be simplified, ribosomal RNA modification, and then this interesting series of correlates that I've tried to illustrate between, say, glycopeptide resistance in enterococci and staphylococci, and potential to detect changes to cell wall components, and possibly even a potential for rapid susceptibility tests. If Bruker are happy, then there's a whole series of PhD studentships for the audience to bid, to supervise, to test all these possibilities, because they do need exploring. Thank you indeed.
Thank you so much, David, for this, like always, furious and inspiring talk, and I'm sure there will be some questions for you in the audience.
Maybe I can start with a question. Thank you, it was really an interesting talk. Do you think we will be able in the future to detect such drug modifications or rRNA modifications directly from the sample itself?
You may stand a chance, for example, with urines where you can get very high densities of bacteria by harvesting the cells. If you're looking where there's a low bacterial density in the blood, I would think not. And there, possibly the DNA-based technology where you've got PCR amplification may stand a better chance.
Any more questions? If not, thank you again, David.
Thank you.